Hello, Pirates. Today's topic is chromosomes. And the essential question that we're focused on is what structures store and transmit genetic information. And chromosomes really are the DNA, those long strands of the double helix, all packaged up into different structures. And as we go through the next couple weeks, we're really focused on how cells divide, and we're very interested in how the genetic information, that DNA, is passed on to the new cells. So new cells that could become regular normal body cells, or cells that might become gamete cells that could reproduce to make offspring. So for your chromosome notes, these are going to go in your notebooks on page 55. And we're just going to go over these basic terms for chromosomes. So these are all terms that mean chromosomes, structures that are containing DNA, but they mean the chromosome in different formats, kind of, okay? So the first one that you guys have on here is just a basic, what is a chromosome? And when we use that term chromosome, instead of using DNA, we're talking about a organized DNA strand, okay? So this is an organized DNA strand. And this organized DNA strand is going to have the DNA supercoiled. And what that means is that the DNA is coiled around a protein called a histone, and then it's coiled again and again and again. So if you look at this picture up here at the top, it's showing you guys how the DNA is super coiled around histones until we can unravel it to make it look like that twisted ladder, the double helix. But zoomed out, it just looks like the chromosome. So this is a super coiled, DNA around histone proteins. Kind of like with Christmas lights right now. So you know how the Christmas lights can be a real mess? If you wrap them around something, you can organize them before you put them away. Okay, kind of like when you buy them, they're organized and wrapped around the cardboard instead of just being a big old mess. They're coiled, and then sometimes they're coiled again and again. They're super coiled. So when we look at the chromosome, it looks like the letter X, and our letter X is just the super coiled DNA. So it's these DNA strands wrapped around proteins called histones, and then supercoiled and supercoiled into these packaged structures that we refer to as chromosomes. Now, the chromatin is when the DNA is relaxed. So instead of being supercoiled and organized, this is when it's all relaxed. And when it's relaxed, it's almost like the pages of the book, the instructions, are able to be read to make proteins. So this is when the DNA is uncoiled DNA strands. And so what this looks like, when the DNA is in the cell like this, it looks like genetic spaghetti. So it's all unwound, kind of like if you just put your Christmas lights away in a mess, all right? Or a ball of yarn when it's all unraveled. An unduplicated chromosome is just a single copy of the chromosome. So right now, if you have cells that are just going out their normal business, they just have a single copy of each chromosome. So it's a single copy of DNA. And like I said, it is not yet copied because the cell hasn't decided that it's going to divide yet. And so a lot of times it'll look like just one single strand. All right, so this right here, for example, this would be like one single unduplicated chromosome. All right, a duplicated chromosome is going to be one that has been copied. So once the cell decides that it's going to divide, it's going to replicate its DNA, and then we're going to have two copies. So this is going to be two identical. So we'll have two identical DNA strands. So instead of just having one strand of this supercoiled DNA, now there are two strands. So if I have my beads right here, I have one, and now I have two. So I went from having an unduplicated chromosome to having a duplicated chromosome. And what you can see with my chromosomes is they're stuck together here in the middle. So once a chromosome is duplicated, they are going to get stuck together. And what they get stuck together with is something called a centromere. So this magnet is acting as the centromere. So the centromere is a structure that holds duplicated chromosomes together. So here I have my original strand. It was not duplicated, and then it gets duplicated. And then here in the center, that centromere is going to hold those strands together. So just like this magnet. So here we have the magnet holding the two strands together. That's the centromere. Now, these unduplicated chromosomes, they can also be referred to as sisters. So sometimes they're referred to as sister chromatids, and sister chromatids are identical. So these are identical, duplicated strands of DNA. So here is one sister, and here is the other. And like I said, they're held together there with the centromere. So here we have one sister and then the other, and like we said, they're held together with the centromere. So here's one sister and here's the other. And like I said, they're identical copies of each other, just being held together. Now, another term with chromosomes is called homologous chromosomes, and the root of that word homo means the same. So homologous chromosomes means that these are the same type of chromosomes. So they're the same type of chromosomes, but they're inherited from each of your parents. So they're the same type of chromosomes from each parent. And the information can be different. Okay, so the info can be different. So here I might have the information from your dad. So here we've got a chromosome from your dad. So this one's coming from dad. There's the centromere holding the copies of that together. And then you're going to inherit genetic information from mom as well. And so these chromosomes are considered to be homologous. They're the same size and shape. They're carrying the same type of information. You know, for example, maybe this one's carrying the gene for eye color right here. And then over here, mom's is also carrying a gene in the same location for eye color. Now, it's the place where eye color might be carried, but the information might be different. Maybe dad's genes are coding for, brown eyes, and maybe mom's genes are coding for eyes that are blue. All right, when cells divide during a process called meiosis, which we'll talk about later on next week, sometimes the homologous chromosomes form what we call a tetrad. So homologous chromosomes form a tetrad. So this happens when homologous chromosomes are bound together. And so when they're bound together as 4 chromatids. So they end up 4 chromatids all stuck together, and tetra means 4. So 4 chromatids are all stuck together. So what happens is your duplicated chromosomes from dad get stuck together with your duplicated chromosomes from mom, and now we have a tetrad. We have 4 chromatids. All right, and this happens during a process called meiosis. And why this is important in meiosis is this is when we have the good old genetic scramble that creates the genetic variation why you can pass on traits from both your mom's side of the family and your dad's. And so what this happens is in crossing over homologous chromosomes, so homologous chromosomes will switch sections, okay? And this is going to create new genetic variations or combinations. So new genetic varieties. And what happens is that sometimes your dad's chromosome gets tangled up with your mom's chromosome. And what happens is that a chunk of your mom's chromosome ends up at the end of your dad's, and a chunk of your dad's ends up on your mom's. And so now we've got chromatids that are eventually going to be sorted into gamete cells that are going to have something new, something different. And you guys can kind of see, like, say these are our homologous chromosomes, say these are from mom, and say that these are from dad. So we've got homologous chromosomes, they're the same size and shape. When they form a tetrad, they all stick together, okay? And what's going to happen is they cross over. So ideally here, I've got the magnets, there we go, is maybe this chunk gets twisted and tangled with this one. And so what happens is now this one might have a little bit of the genetic information from mom. And this can happen in multiple locations. So maybe the genetic information gets scrambled and traded there. And maybe at the top, these get twisted and tangled up, kind of like necklaces in a jewelry box. And they break apart, and then they reattach. And so now, and we'll talk about meiosis next week, what I'm going to have is I've got new combinations. So instead of just yellow, I've got yellow with a little bit of red, or red with mostly yellow, yellow with a little bit of red. I've got red with yellow, and red with yellow in a different gene. So this is what's going to create the genetic variety that's going to be passed into egg cells and sperm cells. So crossing over is really important for genetic variation. So it all happens during crossing over. All right, overall basic facts is that humans have 23 pairs of chromosomes. And you inherit one set of 23 chromosomes from your mom. And you are going to inherit one set of 23 chromosomes from your dad, your biological father, your biological mother. This is what makes them your biological parents. You share biology, you share DNA. And what you're going to have a total of is we're going to have a total of 46 chromosomes. So one set from mom, one set from dad. These chromosomes are divided up into different pairs, which we refer to as a karyotype. Okay, so this is a karyotype. And we're able to diagnose genetic conditions sometimes through these. But what they do is they take a picture of the chromosomes during mitosis when they're all condensed. And we can see the pairs. There's pair number 1, 2, 3, 4, 5, all the way through 23. And you can see there's one that was inherited from mom. So one of these chromosomes came from mom. And you can see they're organized from largest to smallest. There we go. And then the other chromosome is inherited from your dad. And so you are carrying genetic information from both your mom and your dad. And then how those genes get expressed depends on, protein synthesis. But you guys can see here, this person, the 23rd pair is the sex chromosomes. That determines your gender. And if you inherit the genetic combination XY, you are female. And if you're male. Sorry. My bad. If you inherit XY, you are male. If you inherit XX, you are female. Okay? So when we look at this person right here, they have an X and a Y chromosome. So genetically, this person is male. All right. Like I said, we can determine gender by looking at chromosomes. We can also determine other genetic abnormalities. Sometimes people have an extra 21st chromosome, which would result in Down syndrome, or they could have like triple X syndrome. They could have 3 X chromosomes for the 23rd pair. So this karyotype is used for diagnosing genetic disorders. So what you guys are going to do is you guys want to answer the essential question at the bottom. So what structures store and transmit genetic information? Answer that right here. And then for what you're going to submit, you guys want to create a acrostic poem and just write chromosomes down page 54 and then write sentences about chromosomes. I hope you guys are having a great day. Welcome back after break.